So, what happens when you grow stem cells in outer space? The answer could change how we understand aging, immunity, and even disease. Welcome to the weird world of microgravity biology. Hey, it's me, Bruno, and welcome back to Bruno's BioLogs, the place where we dive into all things weird, wonderful, and wildly cool in spatial biology and microscopy. Today we're going to be leaving Earth behind, literally, because we're talking about the new frontier in microscopy and stem cell science, the International Space Station. Scientists are sending human organoids into orbit and analyzing how they grow in space using advanced microscopy. So let's buckle up because we're heading straight into Earth's lower orbit. So why space? Well, the microgravity environment aboard the ISS mimics certain aspects of accelerated aging. So without the pull of gravity, cells behave differently. Their shape changes, gene expression shifts, and their metabolism adapts in strange ways. So studying cells in this environment can help researchers understand what might happen to astronauts on long missions, and also give us clues on how our cells are behaving during aging and disease. Scientists aren't just sending flat cell cultures up anymore. They're sending 3D human organoids, mini versions of brain, gut, kidney, and even heart tissue. These are complex stem cells-derived structures that mimic how real human organs behave. For example, the NIH's Tissue Chips in Space initiative, in partnership with SpaceX and NASA, has sent up heart and immune organoids to study inflammation, degeneration, and aging under space conditions. Fun fact, some of these organoids were developed right here on Earth by groups at UC San Diego and UCSF before being launched aboard Dragon cargo capsules. Okay, so where does microscopy fit in? Well, once these space-grown organoids return to Earth, they're cryopreserved, sectioned, and analyzed using powerful imaging tools. Researchers use confocal microscopy, immunofluorescence, and even spatial transcriptomics to map out how space affected the biology of these organoids. For example, one study showed that gut organoids grown in microgravity had elevated inflammation and altered barrier integrity, and that was only visible using multi-channel IF and 3D confocal imaging. We're not just learning about space travel, we're learning about ourselves. Organoids in space gives us a magnifying lens into biological aging, tissue regeneration, and stress adaptation. And microscopy is what allows us to visualize those changes at a cellular and subcellular level. These insights can inform how we treat age-related diseases, optimize regeneration therapies, and maybe, just maybe, help astronauts stay healthier on missions to Mars and beyond. To me, the coolest part is this: space is acting like this biological pressure cooker. It accelerates changes that would take years on Earth, and we get to see those changes cell by cell with our microscopes. This is a beautiful reminder that great science doesn't always have to happen in high-tech Earth labs. Sometimes they're happening 250 miles above our heads, floating in zero gravity. Hey, thank you so much for floating through space in spatial biology with me today. If you enjoyed this episode, hit a like, subscribe, and drop a rocket in the comments below if you're willing to send your cells into space. I'm Bruno, and I'll see you next time on Earth with another episode of Bruno's BioLogs.